Hello. So I'm going to present today some work we did within work package 5 as well on applying genetic tools for marine monitoring. So most of the work I'm going to present today is being done by Eva Ilagas, PhD student, co-supervised by Angel and myself. When we have an environment and we want to measure its ecological status, usually the way it works in most cases is that we collect a sample from this environment, we bring this sample into the lab, we do a series of experiments or analysis in this sample, and then we derive a number or a measurement of this sample so that we can infer the status. One of the most known benthic indices is the AMBI index, which is based on the analysis of benthic samples to look for species, and depending on the ecological group these species belong to, we can derive a formula and calculate the index. The problem with this index is that it requires lots of time under the microscope or the lens to identify the species, and it requires expertise as well, which is not always available. So we need fast and easy methods that allow us to go from the sample to the value that we want to measure. So the method I'm going to present today has been partially introduced and is based on the principle that one species equals one unique DNA sequences, ideally. So it means that when we find a sequence in a sample, we know which species is in the sample. So there is a database that contains the equivalence between the species and the DNA sequences. So the workflow of the method consists of collecting a sample, extracting the genomic DNA, extracting the barcodes, the genetic IDs from this genomic DNA, and then from those barcodes deriving the species that are present in this sample. The problem is that before this method can be implemented in routine monitoring programs, there are some questions that need to be solved. So for example, when we sample, how much volume should we sample, how much volume is representative of the whole community, which type of sample should we use, water or sediment, for example, which are the target organisms, or which genes should we target, which is for the most suitable barcode, which DNA extraction method, so depending on which method we use, we can get some species that are extracted easier than others, and so on. So during this project, we have been tackling these questions, and we have published a number of papers in which each of these issues have been addressed. So in summary, we have established protocols for laboratory analysis and for bioinformatic analysis, and we have determined the conditions that provide the most accurate metabarcoding-based taxonomic inferences. We have also increased the database in about 250 new barcodes. So basically, we have produced standardized procedures for metabarcoding-based monitoring. We can conclude that macroinvertebrate metabarcoding-based AMBI is then possible, and this is shown in those two figures, which is part of the whole work we did, in which you can see that the taxonomic inferences we get from genetic data are similar to the ones we get with morphological identification, and that the AMBI index derived from metabarcoding data are equivalent to the AMBI index we get from morphological identification. The problem is that this method still has some drawbacks. One is related to the types of samples we need to handle. So sediment samples, they are of different nature, and in order to collect a subsample from this sample that is representative of the whole community is not that easy. So we needed to develop some protocols in which that involve decantation and homogenizing of the samples. Still is quite tedious. It's not like taking a small volume of samples. And also, metabarcoding does not allow quantification, so we cannot know the number of individuals of each species we have in the sample. We have shown that the index works quite well with presence-absence, but still there is room for improvement here. So as a summary, we have the traditional AMBI, which requires short inner identification, which is tedious, and we have what we call the genetic AMBI that is improved in terms of cost effectiveness, but still requires some pre-processing of the sample that is not ideal, and also we cannot infer number of individuals from number of sequence reads. So we thought that developing a microorganism-derived index could be an option as well. So working with microorganisms means easy sample handling because we can work with small volumes. We can sort of quantify in this case, but the problem is that we don't have an index that is developed. So that's what we tried to do. So we defined the groups of bacteria, so similar to what was done for macroinvertebrates in AMBI, based on literature search. So we classified the bacteria into tolerant, related to pollution inputs, or the other ones. And then, similar to the AMBI, we developed this formula. And as you can see here, this new index, this micro CAMBI, correlates quite well with the pressure index that was calculated using a series of different parameters. So this means that the micro CAMBI could be a promising tool to assess environmental status. Also, the good thing about this is that bacteria are ubiquitous, so we can probably use the same groups of bacteria in different parts of the world, and then if they respond rapidly as well. The problem is that it's challenging because the genetic diversity within a single bacterial taxa is huge, so taxonomic identification is tricky and requires validation. So at the end, this is what we have so far that is ready to use, but there is room for improvement, so we can work further in developing the micro-GAMBI, also based on microbial eukaryotes. But ideally, what we would need to do is to identify indicative taxa and genes so that we can develop a kit, and hopefully this kit should be portable so that we could avoid bringing the samples into the lab and go through the sequencing process. But this is not far, or not close, it's not in a near future. And that's it.